Chromatography is an important biophysical technique that enables the separation, identification, and purification of the components of a mixture for qualitative and quantitative analysis. Chromatography is a very useful technique as it allows the separation of components of a mixture on the basis of their nature, structure, size, and other properties. There are two essential components of all chromatography techniques. 1. Mobile phase. 2. Stationary phase. Chromatography, in general, is based on the principle that components of a mixture are separated when the mixture added to a mobile phase is moved through a stationary phase, which mostly is a solid surface, resulting in some components of the mixture being attached to the stationary phase. At the same time, the rest is passed along with the mobile phase. What is a stationary phase? The stationary phase in chromatography is the phase that is either a solid or liquid particle attached to a glass or a metal surface on which the components of the mixture to be separated is absorbed selectively. The stationary phase to be selected for a chromatographic process depends on the nature of the components to be separated and the type of chromatography. Depending on the type of chromatography, gel beads, thin uniform paper, silica, glass, some gases, or even liquid components are used as a stationary phase. What is the mobile phase? The mobile phase in chromatography is the phase that is either liquid or gas that is passed through a chromatographic system where the components of the mixture are separated at different rates by adsorbing them to the stationary phase. The mobile phase is the solvent that carries the mixture as it moves down the stationary phase. The term mobile indicates that the phase is moving down the chromatographic system, whereas the other phase remains stationary. Alcohol, water, acetic acid, acetone, or some gases are the commonly used mobile phase in different chromatographic techniques. Types of chromatography. 1. Affinity chromatography. 2. Anion exchange chromatography. 3. Cation exchange chromatography. 4. Column chromatography. 5. Flash chromatography. 6. Gas chromatography. 7. Size exclusion chromatography. 8. High performance liquid chromatography, HPLC. 9. Hydrophobic interaction chromatography. 10. Ion exchange chromatography. 11. Liquid chromatography. 12. Paper chromatography. 13. Reverse phase chromatography. 14. Thin layer chromatography, TLC.